student from Texas A&M University Kingsville Citrus Center, and she's going to talk about genetic transformation of microtomato tomato with a citrus calcium signal modifier gene, CSM1, targeting resistant calcium. So, yes, we are ready. So, whenever you can just click enter.
Okay, as mentioned previously, I don't know if you remember from the other presentations, that citrus has a very important disease. It's called HLD. In my research, we wanted to see if this one gene, the CSM1 gene, is capable of providing resistance to that HLD. But we can't test HLD in Texas. We have to go to Florida and do our trials there. And that's also a lot of money and time just to go over the long juvenile period that citrus has, which is about 5 to 7 years. To compensate for that, we proposed using microtom tomatoes as a model organism to test if the CSM1 gene will deliver resistance and resistance to Candidiasis liberibacter. But the only problem is that Candidiasis liberibacter, the H. canis, is not actually found in citrus or in tomatoes. So we're going to use a subspecies with this Candidiasis liberibacter, so the same one, to infect our T1 plant to see if we can get any resistance. So after I did the transformation of the CSM1 into the microtom systems, I got various transgenic lines. Those transgenic lines, about 17, I selected 3 of them to go through the inoculation, liberibacter inoculation. But other characteristics that we're also going over to see if there's any changes or long-term changes in by overexpressing CSM1 in tomato plants. The only problems were, or the good problems, if you want to see it, is that some lines had low seed sets. And those low seed sets also corresponded to low pollen viability. Continue on to if the transgenic lines also propose resistance to liberibacter. I did a mean longevity test. And the longevity test was basically done over infecting my T1 plants with liberibacter and seeing how many days it took for those plants to die. The overall results for that is that none of the lines showed any amount of resistance to liberibacter. In this chart you see up here, the wild type survived 79 days, and then the other line, columns 1 through 4, survived 82 days. After doing the mean longevity of those lines, we also tested, we also looked at the expression of CSM1 in our transgenic and wild type. The wild type did not have the CSM1 gene, but it did have small levels of expression of the CSM1, which may mean that there could be a homolog also being expressed. The transgenic lines, called 7-5 and called 10-7, they had higher levels of expression in non-infected, and then also lower levels for the infected lines. And those infected lines, you can correspond to the Candidiasis liberibacter being infected, or having an infection rate and decreasing the CSM1 expression of those transgenic lines. So overall, it doesn't seem like the plant species may be overexpressing, but we don't know if the calcium signal modifier gene is actually corresponding in the tomato plant. Thank you.